Biology with Dr. E.
Next, we review organic compounds. Organic compounds contain carbon, with the exception of carbon dioxide and carbon monoxide, which are inorganic. So what makes carbon unique that living systems depend on it? Firstly, carbon is electroneutral. Because of this, it never gains or loses electrons. It always shares electrons. In addition, carbon has 4 valence shell electrons, enabling it to form 4 covalent bonds with other elements and other carbon atoms. With this unique property, carbon can form different structures, including long chains and rings. The 4 major organic compounds are carbohydrates, lipids, proteins, and nucleic acids. They are generally called macromolecules because they are large and complex, containing thousands of atoms. How are organic molecules formed? Many organic molecules are polymers, which are chain-like molecules made up of small identical or similar units called monomers. So monomers are the building blocks for polymers. Polymers are usually synthesized by dehydration synthesis, and they are broken down by hydrolysis. This table summarizes the different polymers and their monomers. Carbohydrates are made from monosaccharides. An example of a monosaccharide is glucose, and the polymers are polysaccharides. Proteins are made up of amino acids, which are the monomers, and the polymers are called polypeptides, or more simply proteins. Nucleic acids are made up of nucleotides, which are the monomers, and the polymers are DNA deoxyribonucleic acid or RNA ribonucleic acids. Lipids generally do not form polymers, so they are not added to the table. As already mentioned, biological molecules are formed from their monomers by dehydration synthesis and broken down into monomers by hydrolysis reactions. In dehydration synthesis, depicted here by figure A, monomers are joined by removing a hydroxyl group from one monomer and removing a hydrogen atom from another monomer at the site of bond formation. Dehydration synthesis removes a water molecule at the bond site and results in 2 monomers linked by a covalent bond. In hydrolysis, depicted here by figure B, monomers are released by the addition of a water molecule, adding OH to one monomer and H to the other monomer. This reaction adds a water molecule to each bond, which breaks the bond apart and forms 2 monomers. Figure C here illustrates the formation of sucrose by dehydration synthesis and its breakdown by hydrolysis. The monosaccharide glucose is joined to another monosaccharide fructose by dehydration synthesis, and water is released in the process. This reaction produces sucrose, a disaccharide, and water. Then hydrolysis adds a water molecule to each bond, breaking the bond and releasing the monosaccharides, glucose and fructose.